Hi, I am Vidhi, and around 4 weeks ago, a crazy yet innovative idea sparked in my mind. What if AI was merged with gene editing? The reason why I thought of this idea is because scientists have to go through so many steps before they can even attempt a gene edit. First, they locate the mutation in the DNA and determine what type of mutation it is. After that, they search for PAM sites near the mutation, design possible guide RNAs, and check if the mutation falls inside the editing window. They also try to predict editing success and even potential off-targeting effect. And right now, much of this work is done using spreadsheets, different bioinformatics tools, manual sequence inspection, which makes the entire process slow and fragmented. And because of this idea, I built a Python code with an AI integration with the help of ChatGPT. So, as I just recently finished this project, let's take a look at the output. Firstly, the user is prompted to paste a healthy variation of a 20 BP gene sequence consisting of DNA nucleotides. So as you can see here, I pasted a healthy gene sequence and I'll click enter. Then I paste the same gene sequence but the mutated variation with one mutation. Finally, if I click enter again, the AI model would automate a mutation analysis, evaluate CRISPR constraints, and predict editing feasibility. The first part of the code detects the mutation between the healthy DNA sequence and the mutated one. Basically, the program compares the two sequences letter by letter. And DNA is made of 4 nucleotides, which are A, T, C, and G. And based on that, if the program finds a position where the two sequences are different, it records that index or numerical position in the base chains. This is important because the rest of the analysis depends on knowing exactly where the mutation occurs. After the mutation is detected, the program determines which gene editing strategy might be needed to fix it. Basically, there are 3 main strategies used in this model: cytosine-based editing, adenine-based editing, and prime editing. The next part of the program searches for PAM sites in the DNA sequence. Basically, for the Cas9 system, the PAM sequence is usually NGG. Basically, the program scans through DNA sequences and records every location where this pattern appears. If there are no PAM sites near the mutation, CRISPR cannot target that region easily, which lowers the feasibility of gene editing. And once these PAM sites are found, the program basically evaluates the CRISPR context around the mutation. This means checking whether a guide RNA could position the mutation inside the editing window. The editing window is usually between positions 4 and 8 within the guide RNA sequence. This is the region where base editors can actually modify the DNA base. The program calculates where the mutation would fall relative to each possible guide and then counts how many valid guides exist. If the mutation falls outside of the editing window for all guides, base editing will most likely not work. After analyzing the CRISPR context, the program basically calculates a feasibility score. This score ranges from 0 to 1 and represents how likely the edit might be able to work. The score is based on several factors, including the number of PAM sites, the number of valid guide RNAs, and how close the mutation is to the center of the editing window. Mutations closer to the center of the editing window usually have a higher success rate, so they increase the score. Finally, the final layer of the system is the machine learning model. In short, the program uses a random forest classifier trained on example data generated from different DNA sequences. Each example includes features such as the editing strategy, number of PAM sites, number of valid guides, and mutation position. The model learns patterns from this data and predicts whether the editing success level is low, medium, or high. This step basically adds an AI component to the system by using patterns in the data to make predictions.